Hello everyone, welcome back to my channel Bani's Biology. Today we are going to learn one of the most elegant gene regulation system in bacteria, that is tryptophan operon, and also called trypoperon. So the trypoperon is a group of gene that found in E. coli that control the synthesis of enzyme that catalyze the biosynthesis of the amino acid tryptophan. So the in tryptophan operon ultimate goal is the synthesis of tryptophan. So when the cell doesn't have enough tryptophan, that means tryptophan is low in cell, it turns the operon on to make more. But when the tryptophan is abundant, that means tryptophan is high, that operon turn off to save energy. Tryptophan operon is a repressible system or repressible operon because the operon is normally turned on and can be turned off when the end product, that is tryptophan, is present in high amount. So the presence of tryptophan represses its own synthesis. That's why tryptophan operon is a repressible system. And tryptophan operon also called negatively controlled because the gene expression is regulated by repressor protein. So the repressor protein is produced from the regulatory gene, that is trpR, and this repressor protein block the transcription. So in negative control, a repressor protein that prevent the RNA polymerase from transcribing the gene. So the tryptophan operon is an example of negatively repressible operon. So in tryptophan operon is beautifully organized into regulatory region and structural gene arranged in a single transcription unit. Let's discuss the component of the tryptophan operon. So in the tryptophan operon, the regulatory gene is the trpR that produces a repressor protein. So the repressor is the key regulator. When active, it blocks RNA polymerase from transcribing the operon. So in tryptophan operon, the repressor is inactive form, that is called aporepressor. So it cannot bind in the operator by the itself. So the aporepressor is inactive form, but this aporepressor can be active when it bind to the co-repressor. It become active. So here is the co-repressor is the tryptophan. So repressor protein become active when it bind with tryptophan. Then this active repressor bind with the operator and operon is turned off. So in first the operon is turned on when the repressor protein, that is inactive form. So when the inactive repressor bind with the tryptophan, that is co-repressor, and that become active, and then it bind with the operator site, and then this operon is turned off. So this operon is switched off when the product tryptophan is abundant. That's why this operon is called repressible operon. But this regulatory gene located outside the operon, and this gene is not closely linked to tryptophan operon but control it. So next is the promoter, that is denoted by P, and this is the binding site of RNA polymerase, and this RNA polymerase help in the transcription begins. Next is the operator, that is denoted by O, the binding site of active repressor. When the active repressor bind, then the transcription is blocked. Next is the structural gene. So in trypoperon contain 5 structural genes, that is trpE, trpD, trpC, trpB and trpA. So these 5 structural genes encode enzyme, and this enzyme convert the chorismate to tryptophan. So this is the structural gene, that is trpE, trpD, trpC, trpB and trpA. And these 5 structural genes that encode the enzyme and that ultimately convert the tryptophan. So these genes are transcribed together as a one polycystonic mRNA. That means a single mRNA carries the information of all 5 proteins. So this 5 gene produces the enzyme, and those enzymes carry out the biochemical reaction and that synthesize tryptophan. So here trpE gene that produces the enzyme anthranilate synthetase component 1, and that catalyze the first step in tryptophan biosynthesis, and that converted chorismate to anthranilate. And next is the trpD gene that synthesize the anthranilate synthetase component 2, and also called PRA synthetase. PRA, that means phosphoribosal anthranilate synthetase. This enzyme work together with the trpE gene synthesis enzyme to complete the first reaction in the pathway. Then is the trpC that synthesize the PRA isomerase INGP synthetase. PRA that means phosphoribosal anthranilate and INGP that means indole 3-glycerol phosphate that carries out the middle step of the pathway and converting intermediate towards indole, that is INGP, indole 3-glycerol phosphate. Then trpB that encode the enzyme tryptophan synthetase beta. It is the part of the enzyme complex that perform the final step of tryptophan synthesis, and trpE that produces the tryptophan synthetase alpha and work with trpB that convert indole plus serine into tryptophan. So this enzyme carries out the step-by-step reaction and that ultimately produces tryptophan. The cell want to produce all the enzyme for tryptophan biosynthesis only when needed. So that structural gene that encode the enzyme that work together to synthesize tryptophan. But this tryptophan synthesis is controlled by a regulatory region that is called leader sequence. So the leader sequence of the trypoperon is a third regulatory region that is located between the operator region and the structural gene, that is first structural gene, that is trpE, and this leader sequence also called trpL, and this trpL region specify a 162 nucleotide long mRNA leader sequence. So this is the mRNA leader sequence, and this leader sequence encoding the leader peptide and attenuated sequence, and this leader peptide is 14 amino acid long. And this peptide contain two adjacent tryptophan codon, that is UGG and UGG, and these two trip codon allow the ribosome to test whether tryptophan is abundant or low, and this leader peptide act as a sensor and that help the cell measure how much tryptophan is available in cell. So next is the attenuated sequence. The attenuated sequence is a specific RNA sequence that is located in the leader region of the trypoperon and it lies immediately downstream of the leader peptide coding region. The attenuated in the trypoperon that includes 4 distinct RNA sequence region, that is region 1, region 2, region 3 and region 4. So region 1 that is the part of the leader peptide coding region, and region 2 that can pair with the region 1 and region 3. So region 3 pair with the region 2 and region 4, and next region 4 pair with the region 3 and form the terminator hairpin, and these 4 regions that form the alternate secondary structure, that is stem and loop or hairpin structure, and this region is responsible for attenuation. So what is attenuation? Attenuation is a regulatory mechanism that control transcription based on tryptophan availability. So the attenuator can form 2 alternative RNA hairpin, and this determine whether transcription continues or stops. So these 2 alternative hairpin, one is terminator and another is anti-terminator. When region 3 pair with region 4, then form terminator hairpin, and this is the transcription termination hairpin, and this loop is formed when tryptophan level are high, and this is the transcription termination hairpin where region 3 pair with the region 4, and this hairpin is followed by a weak AU ridge region. So the hairpin plus AU ridge region causes RNA polymerase to fall off. So the early transcription termination happened and structural genes not transcribed, and another is anti-terminator hairpin when region 2 pair with region 3, and this loop form when tryptophan level are low. This is the structure where region 2 pair with the region 3, and this structure prevent the terminator hairpin from forming. Terminator hairpin is the when region 3 pair with region 4. Here the terminator hairpin is not formed because of the region 2 pair with the region 3. So RNA polymerase continue into the structural gene and transcription is proceeds, and the enzyme for tryptophan synthesis are made. So now that we understand the component of tryptophan operon, so let's move on to the how the cell actually regulate it. So the expression of trypoperon is regulated by 2 coordinated regulatory mechanisms that work together to sense tryptophan availability and adjust gene expression. So these mechanisms are: one is repression, which control the initiation of transcription. So in repression mechanism, the repression acts as the first layer of regulation by determining whether RNA polymerase can initiate transcription. So in this mechanism, Here trpR, that is regulatory gene that produces inactive repressor protein and also called aporepressor. So when the tryptophan is low, the repressor is inactive and that cannot bind to the operator site, and this inactive repressor is called aporepressor. So RNA polymerase can bind in the promoter site and then transcription is begins. Then the operon is on and gene is expressed. So normally when the tryptophan is low, then the repressor protein is inactive and that cannot bind to the operator site, and then RNA polymerase begins the transcription. But when the tryptophan is high, then tryptophan bind with the repressor protein. So this inactive repressor, that is aporepressor, that become active when it bind to the co-repressor. So here co-repressor is the tryptophan. So when inactive repressor bind with the tryptophan, that is co-repressor, then inactive repressor become active. This is the tryptophan and this is the inactive repressor. When inactive repressor bind with the tryptophan, then it become active repressor, and this active repressor bind in the operator site. Then RNA polymerase cannot bind to the promoter site. Then the transcription is turned off and then operon is turned off. Then tryptophan synthesis is off. So in normal, operon is on and can be turned off when the tryptophan is high. So when the cell need tryptophan, then operon is turned on, and so the enzyme for tryptophan synthesis are produced. But when the cells have enough tryptophan, then the operon is turned off. So this amino acid itself activates the repressor and transcription is blocked. So in the repression process, a repressor protein, this is the repressor protein, shut down tryptophan when the tryptophan is abundant. But the repression responds slowly because it senses tryptophan indirectly through repressor protein. So the cell needs something faster, something that can read tiny tryptophan level in real time. So that rapid and real time control is provided by the attenuation. So it is the second layer of regulation and adjusting how much transcription continue. So the repressor works before the transcription begins, but the attenuation works after transcription has already started. So this process happens in the leader region. This is the leader region and depends on ribosome behavior. This is the ribosome. Because of the in bacteria, transcription and translation are coupled. So that is ribosome begins translating the leader mRNA while RNA polymerase still transcribing. So the attenuation begins when the RNA polymerase cleared the operator and enter the leader region. So as the leader mRNA emerges, this is the leader mRNA emerges by the process of transcription, a ribosome immediately load into it and it start translating the tiny leader peptide. So this is the leader mRNA. So the ribosome now approaches two adjacent UGG codon. So in the leader mRNA, there are present two adjacent UGG codon, that is trp codon, sensor of tryptophan. So the ribosome now approaches these two adjacent UGG codon, that is tryptophan sensor. This is the true starting point of attenuation. So if the tryptophan is abundant, then the ribosome glide across this codon without hesitation. But if tryptophan is low, then the ribosome stall right here. So that single difference is fast moving and stalling decided how the RNA fold. So it decides whether transcription will continue or terminate. So now let's see how the attenuation mechanism behave in two situations, when tryptophan level are high and when tryptophan level are low. When the tryptophan levels are low, then the amount of charged tryptophan tRNA is also low. This charged tryptophan tRNA act as a real-time sensor of tryptophan availability in the cell. So its level determines how fast the ribosome moves through the leader peptide. So here the amount of charged tryptophan tRNA is also low. So as a result, the ribosome stall at this two trp codon. So this is the trp codon UGG and UGG. Here is this is the trp codon UGG and UGG, where the ribosome stall at these two trp codon. Then ribosome physically block the region 1. So region 2 is free because of region 1 cannot pair with the region 2. So region 2 easily pair with the region 3. So region 3 cannot pair with the region 4. So the transcription termination hairpin cannot form. So transcription is terminated when the region 3 pair with region 4. So therefore RNA polymerase continues past the attenuator and enter the trpE gene. So the operon stay on to make more tryptophan. So however, when tryptophan is abundant, then what will happen? So when tryptophan is abundant, then the plenty of charged tryptophan tRNA is available. So the ribosome doesn't stall in the trp codon because ribosome quickly translate to trp codon. So ribosome move quickly and reaches the stop codon. This is the UGA, where the movement of ribosome is stop. So in doing so, the ribosome physically block the region 2. So this is the region 2 site and this is the stop codon where the ribosome movement is stop. So physically region 2 is blocked by the ribosome. So region 2 cannot pair with the region 3. So here region 3 now easily pair with the region 4. So the forming the classic 3-4 pairing that form the transcription termination hairpin. This is the transcription terminator hairpin and this structure destabilize the RNA polymerase at the downstream AU ridge region. This is the AU ridge region and causing transcription to terminate at the attenuator. So transcription stop before the structure gene. So the operon shut down because the cell already have enough tryptophan. So these two systems, that is repression and attenuation, are not redundant. They complement each other. So repression that prevent transcription from starting when tryptophan is abundant and attenuation that adjusts transcription during elongation responding to subtle change in tryptophan level. So together they ensure that the cell produces tryptophan synthesizing enzyme only when they are truly needed and conserving energy and maintaining metabolic balance. So let's see the summary of these two tryptophan mechanism. So when the tryptophan levels are low, then the repressor is inactive. Then ribosome stall in leader peptide. Then form the anti-terminator 2,3 form. Because ribosome stall in leader peptide, then ribosome physically block the region 1. So region 1 cannot pair with the region 2. Then region 2 is free. Then region 2 can easily pair with the region 3. So then transcription is continue. Then operon is turned on. But when the tryptophan levels are high, then tryptophan activates repressor. Here tryptophan act as a co-repressor. Then co-repressor bind with the inactive repressor, that is aporepressor, and then inactive repressor becomes activate. Then this activate repressor binds operator. Then ribosome cannot stall in the leader peptide. Then ribosome moves quickly and then physically block the 2 region. So 2 region cannot pair with the region 3. So region 3 is free. Then region 3 can easily pair with the region 4 and that form the transcription terminator hairpin. Then transcription is stop and operon is off. So the tryptophan operon is a perfect example of how bacteria save energy with smart regulation, with repression acting as the main on-off switch and attenuation adjusting transcription in real time. So the cell produces tryptophan only when it's truly needed, and this dual control makes the operon fast, efficient and incredibly precise. So if this lecture helped you understand the concept, make sure to like, share and subscribe the channel for more clear and simple biology explanation.